So one of the first basic concepts when we talk about the body and the physiology of the body is understanding levels of organization. So our bodies are composed of variety of levels of organization at the very basic, and you can get pretty small with this, okay? But we're going to start at just the chemical level with atoms, molecules, that sort of level, okay? And then above that, we've got the cellular level. So at the chemical level, we've got molecules and chemical reactions that are at the basis of nearly every function of our body. Okay? And so all of the basic chemistry of forming bonds, breaking bonds, enzyme reactions, all of the basic chemistry is underlying nearly every function in our body. Okay? And we'll see that play out as we go through. Then we get to this next level of organization, is the cellular level. And the cellular level is kind of this incredible level of organization where we have these living functional units that make up our body. Okay? And these living functional units are both somewhat autonomous and then at the same time completely not autonomous. Okay? They are individual units living individual lives, but really under the fascist rule of our body. Okay? They're not allowed to do anything without permission. And if they start going rogue, we have a word for that, cancer. Okay? So the cellular level, and in our bodies, our cells are not all the same. We actually have a variety of cell types that have evolved over time, and then those cell types come together and they form tissue types. Okay? And we've got a variety of tissue types, and these tissue types have different functions. Okay? These different cell types come together to make different tissue types, and each tissue type has a particular job. Okay? And then different tissue types come together to form a single organ. Okay? And then those, or an organ system. So skeletal muscle, for example, forms our skeletal muscle system, right? Which is not a discrete organ, it's really an organ system. And then these organs are these incredibly organized structures that have, again, very specific responsibilities. And oftentimes, every single organ in our body has multiple functions. Okay? Functions that you may not even appreciate or realize. You know, we think about the kidney as being, for example, a filtering organ, and it is. The kidney's got a tremendous job of filtering nearly our entire blood volume every minute of our lives. Okay? So, but while it's doing that, that's its day job, and then it's moonlighting by maintaining our red blood cell count. Right? It releases erythropoietin and it maintains red blood cell count. And it's also moonlighting and it's maintaining our blood pressure. Okay? And it's also moonlighting and it's got like 5-6 different moonlighting jobs. And it's also controlling the rate at which we're absorbing calcium. Okay? So the kidney has a tremendous array of functions beyond just being a filtering organ. And it can do that because it's composed of a variety of different tissue types, each with its own particular functions. Okay? So these organs, each individual organ then comes together and creates an organ system. So the kidneys come together along with our bladder and our urinary tract to form our urinary system, renal urinary system. Okay? The cardiovascular system is composed of both our heart and all of our blood vessels. Okay? So these organs come together and they create an organ system. And then the organ system as a whole, each organ system plays a key role in maintaining the entire organism. Okay? And then, as we'll talk about later, the functions of these organs, organ systems, okay, the functioning of these organs and organ systems is important for both the healthy functioning of the cells and also then the healthy functioning of the organism as a whole. Okay? So there's this sort of interdependence of the healthy functioning of the organ systems and the cells. The cells have to work in order for the organs to work, and the organs work to keep the cells healthy. Okay, so we'll talk about that a little bit more. So this, anybody recognize this painting? Alex Gray. He's this like crazy psychedelic, did a lot of acid, I think, and mushrooms in his day. But he is this artist who, I love him because he spent 2 years just hanging out at Harvard Medical School morgue and just learning anatomy, you know. So a lot of his early work, it's just everything is just the anatomical drawings are just so spot on. But then because he's got his own personality, he also layers a different layer of organization on the body. So he's has this incredibly anatomically detailed painting of the body, and then he's added the chakras, right, to it. So he envisions a different layer of organization. So one thing before we go on is on this topic of layers of organization in the body. If you go to the discussion board, I like to, I love media, you know, and I love media and I love art, especially as it pertains to the sciences. And so I will often talk about and post cool things that I find, things that I really enjoy. And I never really had a good place to put it, so I just decided to start this forum on the discussion board called Fun Things to Do, Watch, and or Listen to. And this is, nothing on this is required. Nothing is required, but many of it might be fun, okay? So this one, we'll watch just a tiny bit of it. So there is a growing appreciation for a different level of organization, and that's our human microbiome. That basically there is an entire ecosystem. So you think of the body as having a slightly different level of organization, and that is the microorganisms that inhabit us both on the outside and on the inside. And that basically we are not just, you're not just Bob. I don't know if there's a Bob in the room, but you're also like, you are the ecosystem of Bob, right? And that's a really cool way of thinking about your body. But that basically this microbiome, this collection of non-harmful microorganisms that live in and on you, actually contribute to your health. And that when we do things that disturb it, it creates disease. Okay? And we'll talk about that a lot when we get to patho, especially in the GI tract. So, oh, this, I don't think that the volume is working on this thing. Oh, it is.
And in the desert, each habitat on your body is home to a unique community of microbes.
Can everybody hear? Okay.
To thrive in a particular habitat, organisms must be able to take advantage of resources and cope with environmental challenges. They often have special adaptations that help them survive.
Oh, sorry. Oops.
Creatures in tidal pools have structures that anchor them to rocks, keeping them from being swept away. Similarly, microbes in your mouth use sticky molecules to attach themselves to your teeth. Near tidal pools, a different set of creatures lives on the soft sand of the ocean floor. In your mouth, a unique collection of microbes lives in softer areas too, including your gums, cheeks, and tongue. To support a living community, a habitat needs plenty of resources. In your mouth, both on teeth and on soft tissue, microbes have access to oxygen, water, and nutrients in the form of food particles, skin debris, and proteins secreted in saliva. Your large intestine provides a different set of resources. It is a warm, damp environment where nutrients are plentiful, and it's home to the body's densest and most diverse community of microbes. Like the species in Earth's ecosystems, members of your body's ecosystems depend on one another for survival. Lining the inside of the intestine is a layer of mucus. The microbes living here form a biofilm, where different types of microbes perform special jobs to keep the entire community healthy. Your body's ecosystems are diverse. Although it's a single organ, your skin has many different habitats. Each habitat has a unique combination of resources and challenges, and its own profile of microbes. Your forearm is a dry environment that gets lots of sunlight. Microbes that live here have adaptations to protect against light damage and to keep from drying out. Your underarms are damp, with lots of sweat glands and hair follicles. Just like in the soil, where microbes are most dense around plant roots, microbes on your skin are dense around hair follicles, which contain the roots of your hair. Oily areas, like the skin on your back, tend to have fewer types of microbes. Oil is often antimicrobial, yet some microbes have adaptations that allow them to live here, and some even use the oil as food. Extreme environments on Earth have the fewest species, and those that can live there need special adaptations in order to survive. Your body has extreme environments too. In your stomach, microbes face the challenge of extreme acidity, yet those with the adaptations to survive have two big advantages: access to your food and very little competition. We still have a lot to learn about the ecosystems of the human body, but we do know proper balance is important for our health. Both in Earth's ecosystems and in our bodies, every species is important in keeping the ecosystem healthy. You are your body's wildlife manager.
It also has a really fun song to it. I'm going to be thinking about that song all night. So it's really, and it's really sometimes easy to consider like little movies like that to be kind of contrite. But at some point you're going to take care, if you haven't already as a nurse, at some point you're going to take care of a patient with C. diff, right? Who's treated a patient with C. diff or took care of a patient with? It's really awful, right? So C. diff, C. difficile, is a microorganism that, in a healthy GI tract, could never survive because it's actually not a very robust organism. But in the GI tracts of patients who, let's say, a cystic fibrosis patient who's constantly taking antibiotics because of multiple respiratory infections, that patient doesn't have a healthy ecosystem in their large intestine. And then, so that C. diff doesn't have as much competition. Right? So patients who get C. diff, which is quite devastating, tend to be compromised patients, not healthy patients, with a floral environment in their gut that is abnormal, right? And so there's a treatment a not very popular treatment, but yet effective treatment called fecal transplantation, right? Where you take a fecal sample from a healthy person with a healthy ecosystem in their large intestine, and you clean up the feces, right? You extract all the microorganisms and you insert it into the GI tract of the patient with C. diff. And it's a very effective treatment. But why don't we have widespread fecal transplantations, right, in the hospital? Because it's not very popular, right? But it is a level of organization. There's actually, if you're as old as I am, you remember the Human Genome Project when it was first launched and then all of a sudden completed, and it's like, what? It's finished? A couple years ago, they actually started the Human Microbiome Project to characterize all of the microorganisms that live in and on humans. Yes, ma'am? I don't know the ins and outs of the fecal transplantation procedure, but it's a good question. I don't know. So let's move on. Let's move on. Okay. Okay. So you've probably read this sentence in like 4th grade, right? Cells are the basic units of life. But they are, right? So the cells in our body, while they all tend to, different cell types look different, they all tend to have the same basic structures with all the same basic functions, okay? And so these structures, you guys have studied this before. Basic structures, the most obvious one is the nucleus. It houses the DNA. We've got the rough endoplasmic reticulum extending out from the nucleus. We've got the mitochondria, where we get all our energy in the form of ATP. We have the ribosomes that are either free-floating in the cytosol or attached to the endoplasmic reticulum that are translating and producing proteins. We've got the Golgi complex that are sorting and sorting proteins and sort of sending them off to their particular destinations. We've got our organelles of intracellular digestion, our lysosome and our peroxisome, okay? And they both carry out different chemical reactions, different digestive processes. We'll go in a little more detail as we get through. We've got the cytoskeleton and intracellular structural proteins in the form of microtubules, microfilaments. And then we've got the plasma membrane, which somewhat taken for granted, but it's an essential part of the cell. Anything that damages the plasma membrane can easily kill the cell. So some of these individual organelles, just consider them and sort of remember, to quote a W quote, remember what some of their basic functions are. So the nucleus. The nucleus is the most easily recognizable organelle in a cell, and it's where DNA is housed. So the DNA in the nucleus never leaves the nucleus, right? So the DNA stays in the nucleus, and it houses the instructions for making every protein that we could possibly hope to make. Now, the way we go from instructions for a protein in the form of DNA to actually making the protein is, since the DNA isn't going to leave the nucleus, what we do is we make a copy of the DNA. And we make the copy in the form of what we call messenger RNA. Okay? So we produce messenger RNA that's a copy of a protein within the DNA. That messenger RNA leaves the nucleus and it enters the endoplasmic reticulum. And the nucleus is not only a membrane-bound organelle, it actually has a double membrane. And that outer membrane that membrane is actually continuous with the endoplasmic reticulum. And the space between those two membranes are also continuous with the endoplasmic reticulum. So messenger RNA leaves the inner membrane, enters that space, moves into the endoplasmic reticulum, and finds a ribosome. Okay? And then that messenger RNA is this structure right here. Right? It's a single strand of nucleic acids, and each triple codon, yes, each set of 3 nucleic acids referred to as a codon codes for a single amino acid. Right? You guys all remember that. So this messenger RNA gets to this ribosome, and this is where, so the forming of the messenger RNA we call transcription, and then the reading of the messenger RNA is called translation, basically going from message to protein. And there's actually another thing that I posted that's like a little game where you can actually translate a protein. You too can make a protein. It's a fun little game. I did it right before I came here. It's all I do is I watch YouTube clips in my office and play games, and it's a good job. So what happens is the ribosome moves from codon to codon, okay? And there's so it reads it from codon to codon, and this little blue thing here with this, hexagon attached to it, this is called transfer RNA, where it has the complement nucleic acid sequence to the codon, and that complement, that transfer RNA, is also attached to the amino acid that the codon codes for. Okay? And every protein starts off with that methionine, MET. Okay? That's the start piece for translating a protein. So codon to codon, what happens is the transfer RNA moves over, you go to the next codon, and then the next transfer RNA comes in, and then the amino acid of that transfer RNA attaches to the first amino acid, and so forth. And so codon to codon, that message is read, and the amino acids are strung together, and that protein is formed. Okay? So when we talk about gene expression, what we're really talking about is transcribing the gene, creating a messenger RNA, and then producing a protein. Okay? So whenever we talk about gene expression, we're really talking about creating the protein that gene codes for. Okay? Yes, sir?
What is the ENA?
ENA are just positions on the ribosome. You start off in one position, and then as it's translated, it moves on to the next position. It's you won't have to do that on any exam. It's just an idea. Okay. The mitochondria of every cell is where the bulk of our energy in the form of ATP is formed. Okay? Now, energy is derived from energy substrate. Okay? And for us, energy substrate is a fancy term for food. Okay? So energy substrate comes from 3 sources: protein, polysaccharides or carbohydrates, and fats. Okay? Now, all 3 of these forms of energy substrate are large macromolecules that are composed of individual subunits, right? So proteins are composed of what? Amino acids. Polysaccharides are composed of monosaccharides or sugars, right? Individual monosaccharides include glucose, galactose, fructose. Okay? And then fatty acids are composed of, that's right, free fatty acids, and a monoglyceride, right? So 3 fatty acid chains and a glycerol backbone. So each of these subunits of the energy substrate, okay, which are still really energy substrate, go through 3 basic steps of cellular metabolism. And it depends on the type of substrate. Some bypass different steps. But the 3 steps includes glycolysis, the citric acid cycle or the Krebs cycle, and oxidative phosphorylation, okay? And the electron transfer chain. So glycolysis occurs outside of the mitochondria. If we look at this here, glycolysis occurs outside of the mitochondria, and it does yield some energy, right? And it requires no oxygen. So it's an anaerobic form of metabolism. It produces some ATP, a minimal amount of ATP, and it happens outside of the mitochondria. The Krebs cycle or the citric acid cycle happens within the mitochondria, is also anaerobic, also requires no oxygen, and also produces a minimal amount of ATP. And then the final step, the electron transport chain and the oxidative phosphorylation, does consume oxygen, and it does yield a big wallop of ATP. Okay? So this step in cellular metabolism that occurs in the mitochondria, this is the aerobic step of metabolism. It requires oxygen. And it is actually the only thing we require oxygen for. Right? It's the only thing, just all of our energy. That's all. But this is the step. This is why every cell in our body needs a constant supply of oxygen, so that it can work through its oxidative phosphorylation and produce the vast majority of the ATP it's going to produce. Because that ATP is going to allow the cell to do work, and that work is going to keep the cell alive and keep it functioning and keep the entire system functioning. So mitochondria are very interesting because they're the only organelles in our cells that have something, DNA, right? Mitochondria have their own DNA, mitochondrial DNA. And it only codes for a limited number of proteins, and the vast majority of the proteins that the mitochondria needs comes from the cell's genes, not from the mitochondrial genes, right? But there's a general theory in biology that the mitochondria used to be a single-celled organism out in the world that just decided to live in us, right? We just co-opted it. Why not? It's a good workhorse. And obviously, if that's what it is, we're talking about a pretty mutually beneficial relationship, right? And all of our energy comes from the mitochondria, so that's kind of cool. The lysosomes and the peroxisomes, these are organs of intracellular digestion. So cells are constantly producing proteins and lipids from the inside, and also taking in proteins, lipids, and all sorts of things from the outside. Okay? Now, if just like everybody in your home needs a drain to drain away sewage and everything else, or garbage disposal to get rid of garbage, the cell needs its own sort of incinerator to get rid of waste. And that's what the lysosome and the peroxisome are. But they carry out 2 very different types of reactions, and so they digest different substances. I'm just going to move this because it's getting weird. Okay. The lysosome carries out hydrolysis reactions and generally is responsible for hydrolyzing proteins. Okay? And hydrolysis reactions use hydrolytic enzymes, and it uses water to digest things. The peroxisome carries out oxidation reduction reactions, okay, or oxidation reactions. And the peroxisome is incredibly important for breaking down things like lipids and cholesterol. Okay? And these substances, one of the reasons why we have an entirely separate organelle to carry out these oxidation reactions is that many of the reactions as a byproduct produce hydrogen peroxide, which is absolutely toxic for the cell. So we have enzymes that are designed to neutralize hydrogen peroxide within the peroxisome. Okay? So all the oxidative reactions occur in the peroxisome, and all the hydrolysis reactions happen in the lysosome. Yeah.
You said that lysosome and peroxisomes, they produce hydrogen peroxide?
No. The peroxisomes, the oxidation reactions, the digestion reactions, as a byproduct, hydrogen peroxide is, that's why it's called a peroxisome, right? Hydrogen peroxide is produced, and then there are additional enzymes that neutralize it, so it doesn't kill the cell. Okay? So we have our intracellular digesters. The cytoskeleton. So the cytosol, or the cytoplasm, is not this amorphous blob of free-floating stuff, right? What we actually have is an incredibly elaborate infrastructure of proteins that create what we call the cytoskeleton. And the cytoskeleton, it's sort of drawn here. It's described as sort of the struts and beams of the cell. It gives the cell its particular shape, and it maintains that shape. So a lot of its function is really structural, but also depending on the protein fibers within the cytoskeleton, it's also where it creates a channel for intracellular movement as well. Okay. And the cytoskeleton is composed of really three basic protein fibers. We've got what we call thin filaments, which are also referred to as actin fibers. Thick filaments, referred to as myosin. And intermediate fibers, which are literally just not as thick as myosin and not as thin as actin. Okay? They're just called intermediate fibers. And then, in addition, we also have microtubules that are very different than just a protein fiber. They're really a more elaborate structure. And all of these protein fibers give structure to the cell, but the microtubules also are especially important for intracellular movement, okay? And the movement of proteins and substances to and from, to and fro. Okay? The microtubules also form the spindle fibers when the cell gets ready to divide. Right? So you have all seen those very dramatic pictures of a cell in mitosis, and its chromosomes are getting separated and pulled to the different sides of the cell. And so those filaments are microtubules that are forming what we're calling the spindle fibers that pull the chromosomes to the different daughter cells. Okay? Yes, ma'am. Thin fibers, thick fibers, intermediate fibers, those are the three. Microtubules aren't really fibers. They're a very different type of structure. Yeah. So this picture is visualizing the cytoskeleton of a sample of epithelial tissue called simple squamous cells. These are these very flat, very simple squamous cells. They're the kind of cells, if you've ever done that experiment where you scrape the inside of your cheek and you look on the microscope, those are the cells you're going to see. So there's 3 different visualizations here. So the nuclei are obviously getting dyed in with a blue fluorescence. The green fluorescence is coming from the cytoskeleton, and then the red fluorescence is actually coming from a structure called the extracellular matrix, which is a series of protein fibers that anchors cells within a tissue. Okay. So you can see these cells, they're individual cells with an individual sort of birth, death, and life in between, but they absolutely operate in a collective, right? They are told where to go, how long to live. They're instructed sometimes to kill themselves if necessary. The cells in our body are far more altruistic and cooperative than we will ever be as people. Okay. The plasma membrane plays very important roles for the survival of the cell, and depending on the tissue type, for the function of the tissue and the organ. Okay? So the plasma membrane plays many roles. It obviously defines the boundary of the cell. So it obviously defines the outside versus the inside. It maintains the difference in fluid composition between the inside of the cell and the outside of the cell. Okay? So there is a very important difference in fluid composition inside versus outside of the cell. It maintains, and part of that difference is the difference in ion concentration. Right? And an ion is a charged particle. Okay? Sodium, potassium, bicarbonate, chloride, these are all ions. And these charged particles create electrical gradients, concentration gradients, and they are essential for the function of the cell and the health of the cell. So part of the function of the plasma membrane is to maintain the difference in those ion concentrations. It obviously keeps what's inside in, and it regulates traffic across the boundary. It regulates traffic across the membrane. Okay? So since we have fluid inside of cells, fluid outside of cells, we have well-defined fluid compartments in our body. We have intracellular fluid, which is all of the fluid that is found inside of cells. We have extracellular fluid, which is all of the fluid that is found outside of cells. Okay? And among the extracellular fluid compartment, within the extracellular fluid compartment, we have then subcompartments. Okay? Subcompartments within that. So extracellular fluid includes the plasma, okay, or what's also sometimes called the intravascular space. It also includes the interstitial fluid, which occupies the interstitial space. And the interstitial fluid is the fluid that sits between cells and the vasculature and blood vessels. Okay? There's also a third, very small, third subcompartment of extracellular fluid, and that's what's called transcellular fluid. Okay? And the transcellular fluid is the fluid, it's a very tiny proportion of extracellular fluid, or it should be under normal conditions. It's the fluid that sits entirely surrounded by serous membranes. So that includes things like synovial fluid in our joints constitutes transcellular fluid. The peritoneal fluid in our peritoneal cavity. Okay? The pericardial fluid that fills the pericardium, that membrane that encapsulates the heart. The intrapleural fluid that surrounds each lung. These are our transcellular compartments, and clinically you will hear it described as being the third space. Okay? And there are a variety of pathological conditions that are associated with third space fluid accumulation. Okay? And that's something we talk about in patho quite a lot. But it's when fluid gets stuck, basically, in this third space. Because when fluid enters this third space or accumulates there, it's not easy to get it out. It often requires physical removal to get it out. Okay? But usually that space is minimal, and most of extracellular fluid is found in the form of plasma and interstitial fluid. And then the rest of the body fluid is found inside of cells. So if we take both ICF and ECF, add it together, we get something called total body water, or TWB. Okay? Which, of course, is not pure water. It's a solution. It's a solution with a variety of substances, ions, proteins, other things dissolved in the solution. But if we look at total body water, all of our body fluid, it usually comprises about 60% of total body weight. So of our entire body weight, we are 60% fluid. Okay? Now, that percentage of body weight attributable to total body water varies slightly depending on predominantly body fat percentage. So males and females have different average body fat percentages. Women have a little bit more body fat than men, which means that adipose tissue or fat tissue has less water than muscle. Okay? So that means that women have slightly less total body water than men. And so we have a lower percentage of body weight attributable to total body water. Okay? And then if you look at, obviously, people who are obese versus lean, obese people have a lower total body water and a lower percentage of body weight attributable to total body water. And as we age, somebody told me that many, many years ago, I don't even know if it's still true, but or accurate, that there's something like you basically lose like 2 pounds of lean muscle every year as you get older if you don't do something to counteract it, right? So you get older and you get squishier and you accumulate more fat. And so if you look at elderly people, they will tend to have a higher body fat percentage than somebody even of the same BMI that's younger, because they have less lean muscle. So the elderly will tend to have less total body water, less percentage. And so the significance of this, when someone has lower total body water, they are just a little bit more vulnerable to dehydration. So clinically, that's really the most important aspect. Okay? So as percentage of body fat increases, so does vulnerability to dehydration. So there is a difference in the composition of intracellular fluid versus extracellular fluid. And there's a difference in a wide variety of constituents, or solutes. This is just a little sampling. So, and these are some of the most important ones. So sodium and potassium, number 1 and number 2. Sodium levels and potassium levels, 2 of the most important electrolytes in the body. Sodium concentrations are greater in the extracellular fluid compared to intracellular, and potassium has the opposite pattern, that it's higher inside the cell than out. Okay? Calcium, the concentration of free calcium inside of cells is usually very, very small, and it's highly regulated. It's tightly regulated. Okay? and it's regulated by proton pumps that, calcium pumps that are constantly pumping calcium outside the cell, and also proteins that attach to calcium inside the cell so that it's not free within the intracellular fluid. And the reason why calcium levels are so tightly regulated is that changes in intracellular calcium concentrations trigger reactions in the cell. So you want changes in calcium levels to be intentional and tightly regulated so that you don't trigger sort of a toxic cascade of reactions that kills the cell. Okay? So much higher calcium compared to inside the cell, and inside the cell, very tiny amounts of free calcium. Magnesium, there's a greater amount of magnesium inside than outside. Chloride, greater outside than inside. And chloride levels tend to go along with sodium levels. Okay, so chloride tends to kind of just go along for the ride with sodium. Bicarbonate, greater outside than in, and proteins, much greater inside, obviously, right? The inside of the cell is much higher concentrations of proteins that can't get out of the cell. Yes, sir? No, specific numbers are not important. It's the comparison. Yep. Did you just say it's the girliest thing to ask? Why do women retain more water? I don't know if we do. I don't know if we just like are more conscious of like, oh my God, my belt doesn't fit, right? So I don't necessarily know that we do hold on to more, retain more water than men. Yeah, it's probably the same. But we retain, so the body is not. It has a hard time with excreting sodium loads, so it takes some time for the body to catch up. So if you go, and you would be amazed at the levels of sodium in especially food that you eat that's prepared professionally, like you go to a restaurant. So you can actually cause yourself to hold on to a tremendous amount of water, like up to like 5 or 6 pounds of water. From one really salt-laden meal, right? And it'll take you a good day or two to get rid of that water. So I'm not so sure that women are more vulnerable to that. Okay, we can move on a little bit, and then we'll keep those questions in mind, and I'll solicit some in a little bit. So where we left off, we were talking about the difference in intracellular versus extracellular composition. The reason why the cell can help to maintain this difference in intracellular versus extracellular composition, in part, the reason is because of the composition of the plasma membrane itself. Okay? So if we look at the composition of the plasma membrane, we see that there are 3 main components. We've got structural lipids, we have proteins, and we have some carbohydrate chains that are associated with the membrane lipids and the membrane proteins. So the vast majority of the plasma membrane, the vast majority of it is composed of structural lipids, predominantly phospholipids, okay? And then some cholesterol as well. Okay. Now, these phospholipids and the cholesterol there's a particular chemical property that these molecules have that make it very well suited for forming the plasma membrane. And the property that cholesterol and phospholipid have is that we call these molecules amphipathic. Okay? And something that is amphipathic is a molecule that is both hydrophilic and hydrophobic, where one end of the molecule is hydrophilic and one end is hydrophobic. So something that is hydrophilic, breaking that down, right, it is water loving, so to speak. And what that really means is that something that is hydrophilic is polar or charged. Okay? Something that is hydrophilic is polar or charged, and therefore it can dissolve well in water. It's water soluble. Okay? So a couple of terms that are synonymous with each other. Okay? Hydrophilic, polar, charged, okay, and water soluble. Now, when something is hydrophilic, polar, charged, water soluble, it means that it can dissolve in water. And so water, the structure of water, H2O, looks a little like this. And the structure of water is polar in nature. Okay? So oxygen is bonded to 2 hydrogen atoms, and the nature of a bond, of course, is that 2 atoms are sharing electrons, correct? Now, not all bonds between 2 atoms, 2 different atoms, share electrons equally. So in this relationship between hydrogen and the 2 oxygen and the 2 hydrogens, oxygen hogs the electrons. So the electrons that are shared with these 2 hydrogens spend more time around oxygen than they do around hydrogen. And because of that, this end of the molecule has a slightly negative charge to it, and the other end of the molecule has a slightly positive charge to it. Okay? So when something that is hydrophilic polar charged gets introduced into the solution, okay, like sodium, sodium is going to interact very well with the negative end of oxygen, okay, the negative end of water along the oxygen, okay, and then when you add salt to a solution, it goes from NaCl, right, a solid salt, it hits the solution and it ionizes, it dissociates, right? So you get sodium gets attracted to the oxygen, and chloride gets attracted to the hydrogens. So NaCl, or sodium and chloride, those ions, they are charged, which means that they are water soluble and hydrophilic. The things that are hydrophobic, okay, things that are hydrophobic water fearing or just not liking are things that are nonpolar. Okay, things that are nonpolar, they are not charged, and they are not water soluble. And if they're not water soluble, what's another term we sometimes call them? Fat soluble. I hear some people saying it, right? If something is not water soluble, it's fat soluble. So fat is oil, fat is not charged, right? Those molecules are not charged. So it gets into a water solution and it can't dissolve well. So what happens is that you get what's called hydrophobic interactions, which just means that everything that is fat soluble tries to bunch together and get away from the water. Okay? So your salad dressing, right, always forms 2 layers. No matter how much you shake it, it's always going to form the 2 layers, because the oil layer is not going to dissolve in the water vinegar layer. So a phospholipid and a molecule of cholesterol, those structures are amphipathic, which means that one end is water soluble and hydrophilic, likes water, no problem. The other end is hydrophobic, does not want to have anything to do with the water. Okay? And so a phospholipid is usually drawn in this sort of cartoon circle with two little tails, okay? Where the circle represents the phosphate head that is hydrophilic, okay? And the tails represent fatty acid chains that are hydrophobic. So as you can see, this is a model of the plasma membrane that shows these phospholipids, these cartoon phospholipids, forming a bilayer where the phosphate heads, there's one layer on the extracellular face where the phosphate heads are oriented towards the extracellular fluid. And then the second layer of phospholipids, the phosphate heads are oriented facing the intracellular fluid. And those hydrophobic tails are pointing towards each other. Okay? So phospholipids come in a variety of types. Okay? The most common type is this phosphatidyltholamine. This one, which you don't have to memorize the name at all, phospholipid is fine. But this one's the most abundant that you find in most plasma membranes. But there's actually a wide variety of different types of phospholipids with slightly different structures. Here's the structure of cholesterol. Okay? It has an OH group at one end. That's the charged end. Okay? And the rest is nonpolar. Now, an interesting thing happens when you take phospholipids. You take some phospholipids and you add them to some water. Okay, a small amount of phospholipids and add them to some water. And they naturally form a monolayer on the surface of the water where their phosphate heads are pointing towards the water and their fatty acid tails are pointing out. Okay? You add more phospholipids to the solution, and those phospholipids will naturally, without any coaxing, okay, they will naturally form a bilayer. Okay, when there are more phospholipids available, they will form a bilayer, and it will look like that. So this is a natural orientation for these structural lipids, and it forms the basis of the plasma membrane. Okay? And then interspersed among the phospholipids are molecules of cholesterol. Okay? And they orient themselves very similarly. They orient their hydrophilic ends towards the fluid sides and their hydrophobic ends towards the center of the membrane. Okay? Now, the role that cholesterol plays in the plasma membrane is that at the concentrations that cholesterol is present in the plasma membrane, it adds fluidity and flexibility to the plasma membrane. Okay? Which is an interesting thing because cholesterol is a very rigid structure. Okay? But because most of the cholesterol is oriented in between those fatty acid tails, what it does is it prevents those fatty acid tails from crystallizing with each other and becoming more rigid. Okay? If you've ever thought about the different fats that we consume dietarily, the difference between an olive oil and butter, for example, right? Olive oil and butter. Olive oil at room temperature is a liquid, right? And butter at room temperature is a solid. The reason why butter at room temperature is a solid is because all of the fatty acid chains look exactly alike and they crystallize with each other. And they form this very rigid structure. While the olive oil, there's more double bonds in those fatty acids and they're more kinked, and so they don't crystallize with each other. And so they stay a liquid at room temperature. So those cholesterol molecules are interrupting this sort of solidification between the fatty acid chains, and it keeps the plasma membrane more fluid. So the structural lipids. Now, the other thing about the structural lipids, you notice that if we took we're looking at a cross-section of this plasma membrane, the vast majority, in those phosphate heads, by the way, the hydrophilic portion of those phosphate heads are greatly exaggerated in this cartoon form. Okay? It's really a very tiny part of the structure. It's really this top part, this negative charge, this little positive charge. This is the charged part. So the vast majority of the thickness of the plasma membrane is composed of fat-soluble fatty acid chains. Okay? So that means that the plasma membrane creates a boundary for the cell that is excellent at keeping out water-soluble substances. Okay? A water-soluble substance isn't going to be able to passively diffuse through that membrane. But a fat-soluble substance is going to be able to passively diffuse across that membrane. Okay? Because the bulk of the thickness of the membrane is fat-soluble. Okay? So you think of this in terms of therapeutic drugs. Any drug whose site of action that it resides in the brain, any drug whose site of action that resides in the brain, if you are, let's say, consuming it orally, taking an antidepressant, you're taking a whatever, right? Anticonvulsant, anything where the site of action is the brain, that drug is highly fat-soluble because it has to pass through the blood-brain barrier. Okay? And the only drug that's going to pass through the blood-brain barrier is a fat-soluble drug because the blood-brain barrier is made up of some of the same structures as all other biological membranes. Okay? Yes, ma'am. There's vast array of water-soluble drugs. It's not so. It's very complicated. We'll talk about it. Actually, if you're an ETPer, you're going to get me again. On Friday, I'm going to talk about pharmacokinetics in your pharmacology class. It's my one little guest appearance. And we'll talk a little bit about how drugs that are water-soluble get places. Okay? Proteins. We also see proteins embedded in the plasma membrane. Now, some proteins are, span the entire thickness of the plasma membrane. Some are associated with the extracellular face, the intracellular face, and membrane proteins carry out nearly every function that the plasma membrane, that's critical for the plasma membrane. Okay? So proteins form surface receptors, they form channels, they form, they're involved in intracellular signaling cascades that trigger intracellular reactions, okay, that are absolutely important for responding to things like hormones and drugs. Okay? So proteins do it all. And so there's a wide variety of proteins that we see. This one is a channel. This could be an ion channel, right, that allows the movement of some charged ion across the membrane. This could be some receptor. Some of these proteins have carbohydrate chains attached to them. Okay? You can see some are associated with the extracellular face, intracellular face, and some span the entire thickness of the membrane. So let's take a little side trip and talk just a little bit about proteins. Because if you think about this, your entire genome codes for proteins, right? genes code for proteins. So why are proteins so important? One word. Why are proteins so important? What's the word? What category of proteins in your body could you not live about 10 milliseconds without? Enzymes, right? Every single reaction that occurs in your body is catalyzed by an enzyme. Right? And an enzyme is a protein that makes a reaction happen more quickly. Okay? So if you think back to like high school chemistry and you think about the activation energy for a reaction, okay? So if you allowed a reaction, a normal reaction, to happen without an enzyme, it might take like 5 years to happen, right? But then you introduce an enzyme, and what the enzyme does, this is an analogy, this is not what the enzyme actually does, right? It takes the first thing and it takes the second thing and it's like, all right, do it, okay? It speeds it up. It allows the reaction to overcome that activation energy, okay? It's the matchmaker. You want to meet your soulmate? That enzyme's the matchmaker, okay? So instead of it taking you 30 years, it's going to take you $200 in a week. Maybe. So every single reaction in your body requires an enzyme. And so much of what the genome is coding for are enzymes that are going to create, going to catalyze reactions and create function. Okay? So enzymes are proteins. We also have a whole host of structural proteins, right, which are absolutely critical. So proteins are made up of amino acids, and every amino acid has the same structure. So an amino acid has an amino group with a nitrogen-containing amino group, a carbon-containing carboxyl group, a hydrogen, a central carbon, and this side chain, which sometimes we call an R group. Okay? So every amino acid has this basic structure, and the only thing that they differ in is this R group, is this side chain. And there's only a limited number of side chains that you can have. So we have about 20 possible amino acids, 20 possible side chains. So these are the particular side chains that are attached to that basic structure. And when we start to string one amino acid to the next, they attach by their amino and carboxyl groups. So they're going to form what's called a peptide bond, and then water is liberated. That's the reaction. Okay? So you form a peptide bond, a carboxyl group attaches to an amino group, water is liberated, and now you have a two amino acid polypeptide strand, okay? And so forth as it gets longer and longer. And they are going to be composed of these 20 different amino acids with their 20 different side chains. So there's a trick here, though, because these different side chains have different chemical properties. Okay? So some side chains are nonpolar and hydrophobic, and some side chains are polar. Okay? Some side chains are polar and acidic. Some side chains are polar and basic. Okay? Some side chains have sulfur. Okay? So what that means is when you start stringing amino acids together, neighboring side chains are going to start interacting with each other. Okay? And it's going to start causing the protein to take on a shape. So when we talk about protein structure, the structure of a protein determines its function. The structure of a protein determines its function. Structure equals function. Okay? Now, a protein has many different levels of structure. It has a primary structure, which is literally just the sequence of amino acids. Okay? And the primary structure is the only thing that's actually being directly coded for in that gene. But the primary structure is the basis for all other levels of structure. So depending on the identity and the composition of these amino acids, now these neighboring amino acids start to interact with each other. And these interactions are what we call non-covalent interactions, which means they're not like covalent bonds where they're stuck to each other and they're sharing electrons, right? Non-covalent interactions are things like, I just like you. I just want to spend more time right here with you, right? So you have like, and that's like hydrogen bonding. They're getting close to each other. You're just a little bit negative. I'm just a little positive. And I just like being here, okay? Hydrophobic interactions, okay? We both don't like the rest of them, so let's just stay together, okay? That's hydrophobic interactions. So they start interacting with each other, and they start taking on a secondary structure. And the secondary structure tends to fall under two possible things. They tend to either form a helix, okay, these helices, and also we have these beta-pleated sheets. So we call them alpha helices and beta-pleated sheets. So these like pleats in your skirts and the little ringlets in your hair, right? Those are the two variations in secondary structure. Once it forms that secondary structure, now a whole other set of side chains become exposed to each other. And you get the whole thing all over again. Some are attracted to each other, some are repelled. You do see some covalent bonds. There's covalent bonds in the form of what's called disulfide bonds. So there's a sulfur-containing amino acid here somewhere. I forget the name of it. Methionine. If you get two methionines at different ends of the protein structure, they're going to find each other and covalently bond. Okay? It's one of the reasons why curly hair is so curly, are these disulfide bonds. And if you're like me and you've ever tried to darken lovely yourself at age 12, right? What you're really doing is breaking those disulfide bonds, right? So that you don't have the same curl, breaking those covalent bonds to flatten out the hair. So those interactions, again, more non-covalent, mostly non-covalent interactions, are going to form the tertiary structure, which is really sort of the protein folding in on itself. Okay? The protein folding in on itself. And it forms what we call the tertiary structure of the protein. Then, so a gene might actually code for different subunits, right? And then once they form their tertiary structure, these different subunits start to connect with each other and form what's called the quaternary structure. Okay? So for example, hemoglobin is a 4 subunit structure. So its quaternary structure is 3 subunits that are covalently bonded to each other. Okay? So this tertiary and quaternary structure, the structure of proteins equal the function. It determines the function. Okay? So these proteins in the membrane that form a channel, the only reason why they're forming a channel is because that happens to be their final structure, which is literally defining its function. Okay? And that final structure and function was ultimately determined by that primary, by the sequence, by that primary structure. So we go back to this protein, hemoglobin. which just so happens to be my favorite protein in the whole wide world, for a very good reason. Hemoglobin is a massive protein, right? Huge sequence of amino acids that form the ultimate structure of hemoglobin. Sickle cell disease, right, which is caused by the formation of what we call sickle cell hemoglobin, is caused by a mutation that replaces a single amino acid in the hemoglobin structure. Just a single amino acid is replaced. And that single exchange of an amino acid creates the entire basis for a very debilitating disease. Right? And it completely changes the properties of this hemoglobin molecule. And it especially changes the properties of hemoglobin in its deoxyhemoglobin form when it's not attached to oxygen. The protein just completely changes shape. It starts clumping together, and it causes the red blood cells to form that sickle shape. And then that starts to get clumped together in the blood vessels, and it causes occlusions, and it causes massive pain and thrombosis and really a constellation of problems that equals a very debilitating disease. One amino acid. Okay? So it's really dramatic. And the other thing, one more thing that I'll say about proteins before I move on, is that proteins, their particular structure, is also highly sensitive to the acidity of the environment and the temperature. Because the acidity and the temperature are going to greatly interrupt or interfere with the interactions of these side chains with one another. Okay? So it's going to change the interactions, which is going to change the structure, and it's going to change the function. And we call that denaturing of a protein. Okay? I denature proteins every morning. I love scrambled eggs, right? But that's what I'm doing. I'm denaturing that protein by exposing it to high heat. The two parameters in your body that have the narrowest window of control is body temperature and pH, right? Nearly every other parameter in your body has some fairly wide margin of normal. Blood pressure has a fairly wide margin of normal. But pH, very, very narrow. Temperature, very, very narrow. And it's because of these proteins that are highly sensitive to those two things. Okay? Somebody had it. Yeah. There are a few examples of covalent bonds in the, so all of the primary structure is covalent bonds. The peptide bonds are all covalent. And then there are some examples of covalent bonds like the disulfide bonds, but the vast majority of, like the quaternary structure is almost all covalent bonds. But the vast majority of the time, it's non-covalent interactions. Yeah. Yes, ma'am. So it's not a hydrophobic bond, right? But what it is, so for example, you've got this channel, okay? The channel, the part of the protein that spans the plasma membrane are predominantly going to be composed of amino acids that have hydrophobic side chains. Okay? Hydrophobic interactions are interactions that try to group together hydrophobic compounds and exclude water. Right? So hydrophobic interactions are basically interactions between hydrophobic side chains that try to hide away from the water. Okay? Yes, ma'am. Say covalent bonds? They're much more, so no bonds are necessarily permanent, right? They're all potentially susceptible to interaction with other things. But they're much more stable than the non-covalent interactions. Right, the hydrophobic interactions, hydrogen bonding, those are much less stable than a covalent bond. Hydrophilic bonds, I've never heard it quite described that way, but that would be hydrogen bonding would be an example of that. Yeah. Okay, let's keep going. All right, it's cool stuff. Proteins are awesome. There's actually, I think I put up a little like YouTube movie that sort of goes through some of this protein structure and function if you want to relive it with a very sophisticated British accent. But as I was trying to look for a good animation that shows protein folding, and I came across this. It's just really just like the ultimate nerd game in the world. It's called Fold It. Has anybody heard of this before? But it's literally a computer game that's specific for people who study protein structure, okay? That you are trying to predict the direction that the protein's going to fold based on what you know about the sequence of the amino acids. And so it's all trying to sort of crowdsource knowledge or brain power in order to solve the problem of trying to characterize the protein structure of different proteins. Because there's lots of proteins we still haven't completely worked out the structure for. But I am not qualified for that game at all. But it was very neat. So also within the plasma membrane are some carbohydrate chains that are attached to proteins and lipids. And so when they are attached to those structures, we call them glycoproteins when they're attached to proteins, glycolipids when they're attached to lipids. And especially in the case of glycoproteins, because there are many more glycoproteins than there are glycolipids, oftentimes these carbohydrate chains are especially important in recognition and identity. Okay? So these carbohydrate chains attached to these proteins often are important for attachment, are important for signaling to the immune system and signaling self versus non-self. So often they are involved in recognition and identity. Okay? Predominantly glycoprotein, yeah. Predominantly glycoprotein. Okay. So the basis so the plasma membrane is universally described as being a selectively permeable membrane, okay? Meaning that some things can get through, some things can't get through, okay? And the basis for which how things can cross the membrane, the basis for that is the structure of the membrane. Okay? That it is predominantly a lipid membrane. So in general, things that are hydrophobic, that are fat soluble, have a fairly easy time crossing the membrane, and things that are water soluble have a harder time passively moving across the membrane. Okay, that's the general rule. So the things that determine the ease with which something can cross the membrane is lipid solubility is number one. Okay, and then also size and what particular substance you're talking about. So things that are polar but small, okay? Things like CO2, for example. CO2 has a polar structure. CO2 has some degree of ease in crossing the plasma membrane, okay? Because the polarity is not as strong as some other polar structures. Oxygen is actually a fairly nonpolar structure, right? Molecular oxygen are two oxygen atoms double bonded to each other. So oxygen's going to be forced to share those electrons pretty equally. So that's a fairly nonpolar structure. So oxygen can get cross membranes very easily. Thank God, right? Fatty acids, they're fat soluble. They can cross membranes pretty easily. Small polar substances, okay? There are some passive channels in the membrane that allow small polar substances to cross, okay? And some of these protein channels are selective for certain things. So there actually are some passive channels that allow sodium to enter the cell, passive channels that allow potassium to leave the cell, okay? Passive channels that allow other charged particles to get in and out. But the density of these passive channels really depends on the cell type you're talking about, okay? Big variety depending on what cell type you're talking about. And then water is a very particular situation, okay? Water can generally freely cross the membrane, but it can only passively move across the membrane in limited amounts, okay? Because water actually has a fairly polar structure. So water can pass through the membrane in limited amounts, but most cells in our body have abundant what are called aquaporins, or water channels, that allow water to cross the membrane freely, okay? And these aquaporins, this is actually the structure of a particular subtype of aquaporin. The different subunits are color-coded, okay? So it's a 4-subunit structure. It sits in the membrane. So you notice that these, the part of the protein that's sitting in the membrane look like they're just a bunch of barrels, right? Those barrels represent those helices, right? Those alpha helix, that alpha helix structure. And when you have a protein that spans the plasma membrane, the majority of the portion of the protein that sits in the membrane tends to form multiple alpha helices, okay? So talk about structure equaling function, okay? So you have this very elaborate protein with these this hydrophobic core sitting in the membrane, and then it just forms a little hole, little channel that allows water to pass through, okay? And aquaporins allow water to freely shift in or out of the cell. So generally speaking, unless you're talking about a very specific cell type, okay? And when we get to the renal system, we'll talk about cells in the nephron, that some cells, their plasma membranes don't have aquaporins, okay? But your generic run-of-the-mill cell, plenty of aquaporins, and water can freely move in or out. So when substances are crossing the membrane, they tend to move either passively or they move actively. And when something moves passively, it doesn't require any input of energy, right? It's analogous to something rolling down a hill. All it needs is opportunity. It doesn't need energy. It just needs opportunity to roll down the hill, okay? When something is being transported actively, you have to put energy in because it's analogous to pushing the boulder uphill, right? The boulder, even if it has opportunity, boulders don't spontaneously move uphill, right? You got to really get your elbow grease? No. Energy in there and really push that boulder up, okay? So the same thing with crossing the membrane. So passive transport, or the movement sort of downhill, is in the form of either simple diffusion, facilitated diffusion, or osmosis. Okay? And then active forces, we've got two flavors of active transport. We have primary active transport, which is very straightforward. We're using ATP and we're moving something uphill. Okay? Secondary active transport, it's a little less straightforward. We're actually using a slightly non-intuitive form of energy, not ATP, to move something uphill. Okay? So passive forces, simple diffusion. First off, the foundation, so cell transport, I got a bunch of interesting things in this one. So first of all, even though it doesn't look like it, everything is in motion, right? So when we look, when we consider matter has three states, right? Gas, liquid, solid. Even things that are in a solid, right? The atoms that make up my coffee cup here, they don't look like it. I'm talking about the liquid inside. I'm talking about the actual cup. They don't look like it, but they are in motion. Okay? They're not in a lot of motion, but they're in some motion. You look at the liquid inside, the liquid state, there's much more motion. Okay? And the motion is called Brownian motion, and it's a very random motion. So if those red dots were the water, okay, and the big blue dot was a solute dissolved in the water, this is what my cup of coffee looks like, right? You've got the water constantly moving, you've got the solute along for the ride. That's Brownian motion. Okay? And the gas state, right, if I boil my coffee and all of a sudden some of it turns into vapor, the gas state has even more degree of motion than the liquid state. Okay? So considering that everything is in motion like that, the Brownian motion, the idea behind diffusion is that things are going to naturally tend to move from an area of high concentration to an area of low concentration. So molecules that are more tightly packed together, they're going to collide into each other, and then slowly they're going to take up more space. They're going to diffuse into the area of lower concentration. So you've got two sides of a container separated by a semi-permeable membrane. Okay? The Brownian motion here is creating a tendency towards movement to the right, and the Brownian motion here is going to create a tendency for movement to the left. But since the concentration on the right is so low, that movement to the left is not very much, right? It's certainly not compared to the movement to the right. So you get a net diffusion to the right until you get an equal distribution on both sides. Okay? So now the tendency for movement in each direction is equal. So diffusion requires a concentration gradient. Okay? And it's always trying to get to equal on both sides. Okay? So that's the crux of diffusion. And diffusion, the rate of diffusion can be altered or increased or determined by a variety of things. So there's something called Fick's law. Very smart man, Mr. Fick. He came up with several factors that are going to determine the rate of diffusion. The first is the magnitude of the concentration gradient. Okay? the higher the high is compared to the low, the steeper the gradient, the faster the rate of diffusion. Okay? And as that gradient, as the diffusion proceeds and that gradient starts to get smaller, the rate of diffusion starts to go down. Okay? Another one is the permeability of the membrane. Okay? As if you have a more permeable membrane, you have faster rate of diffusion. Third is the size or weight of the substance diffusing. Something that's smaller, lighter, is going to diffuse faster. The next is the thickness of the diffusion gradient. The thinner, or the thinness of the diffusion gradient. The thinner the diffusion, not gradient, the diffusion barrier, excuse me. Okay? The thinner the diffusion barrier is, the faster the rate of diffusion. Okay? Thinner barrier, it gets across faster. This one right here.
And has that difference in the permeability of the membrane?
It's different because you can have two membranes that are of equal thickness. One can be more permeable to the substance. It's going to diffuse faster compared to the other. They're very similar, but slightly different. Another way of saying the thickness of the barrier is also the diffusion distance. Okay? Whether it's a barrier or it's just distance. Okay? And that becomes very important because you'd be surprised how many absolutely critical processes to survival are accomplished through simple diffusion. Okay? How does oxygen get from your lungs into your blood? Diffusion. How does oxygen get from your blood into your tissues? Diffusion. Okay? So at those sites of oxygen exchange, you'll find a very, very small diffusion distance, very thin barrier, very, very permeable. Okay? You'll find the concentration gradient to be fairly high. And this last one is a huge mechanism for maximizing diffusion in the body. This is the surface area available for diffusion. So you can see we've got this barrier, but not all of it is available for diffusion. It's just this one little area. The greater the surface area available for diffusion, the faster diffusion happens. Okay? And so everywhere in the body where diffusion, simple diffusion is the primary mechanism for movement, you see this tremendous maximization of surface area. Okay? And we talk about that in the lungs, we talk about that in the capillary beds, we talk about that in the GI tract with absorption of food. Okay? So the other thing that affects diffusion, which in the body is fairly stable, but it ties in this idea of the Brownian motion, is temperature. Okay? In the body, hopefully your temperature is stable. You're always approximately 98.6. But so I discovered this guy's videos. I hope this doesn't offend anybody. He's called like the Crazy Russians Life Hacks. Anyway, I'm just going to play it. But he illustrates, it's a pretty...
This little microprocessor makes lives everywhere.
Excuse me, 1 second. Okay. It's actually some pretty cool videos. He's got some really neat things that he shows. But this is a fairly classic experiment that shows, illustrates diffusion and really shows the mechanism of how Brownian motion is really the driving mechanism for simple diffusion. So let's just watch this for like 1 minute, I think.
What's up, everybody? Welcome back to my laboratory.
tonicity is number one priority. In this experiment, I'm going to demonstrate Brownian motion. Let's check it out. So I got here hot and cold water, and I got here ink. I got it from the pen and mixed it a little bit with water. First, we're going to drop a couple of drops of ink in the cold water and then hot water. So just a small drop. Well, it was a little bit more than one drop. As you can see, everything kind of stays together and sinking slowly down into the glass, kind of slowly. Now let's start the same thing in the hot water. Just a small drop. Put 3 drops. Let's see what happens. As you can see in this glass, it's actually kind of sink down really fast. And as you can see, it's actually dissolves before it's hitting the bottom of the glass. And in this glass...
So cold water, hot water, right? As the temperature goes up, the Brownian motion increases. So that higher Brownian motion causes the rate of diffusion to accelerate, okay? Because it's going to distribute those solutes much, much faster. Okay? But as I said, in the body, that's not such a it's not such a relevant thing in terms of Fick's law because body temperature is stable. Okay? But it just illustrates the mechanism of Brownian motion in diffusion. Okay. Yes?
How does having a fever, does that affect?
Having a fever, your body temperature doesn't increase too much, yeah. But it does, increases in body temperature will affect the speed of nerve conduction. It does affect your body, but it's not going to have such a dramatic effect on diffusion unless you're getting into very, very high fevers. Okay, so simple diffusion, it means that the substance can just passively cross the membrane, and it doesn't. All it requires is a concentration gradient. So the sorts of things that passively cross the membrane are fat-soluble substances and water-soluble substances that have an open channel, basically, that have a passive channel for crossing. Okay? And including water, because it has those aquaporins. There are some substances that diffuse across the membrane, but it's assisted diffusion, okay, or facilitated diffusion, meaning there's a protein at the membrane that the substance actually binds to, and the protein moves it across the membrane. Okay? But it's still considered diffusion because it's still only going to move that substance from an area of high concentration to low. Okay? It's not employing any energy. And the facilitated diffusion, some sources include non-gated channels, like ion channels in facilitated diffusion, and gated channels. Okay, some include them, some don't. But most of the facilitated diffusion, examples of facilitated diffusion, tend to be in the form of carrier proteins. Okay? And in the case of carrier proteins, the protein structure is such that the binding site for the substance is either facing one direction or facing potentially both directions, but it's only going to carry the substance from an area of high to low. So this protein is sitting in the membrane. When the substance binds at the binding site, it changes the structure of the protein. Okay? And it changes the structure of the protein so that the protein is now facing the other side. And as soon as that structural change happens, it loses its affinity for the substance. And so the substance is let go. And as soon as it lets it go, it goes back to its original shape. Okay? So with carrier proteins, the only thing that's necessary is that there's a gradient, there's a concentration gradient, and that the substance has to bind to the binding site. So it's still simple diffusion. So an example of a substance that crosses most membranes this way is glucose. Okay? Glucose transporters are facilitated diffusion carriers. They bind to glucose and they carry glucose across the membrane down its concentration gradient. Okay? Yes, ma'am. What do you mean?
How would it know that one side has a higher concentration versus another?
It's more a function of probability, right? That the likelihood of binding is greater at the end with the higher concentration. The likelihood that the substance binds to the protein, yeah, is higher in the area of higher concentration. Yeah, absolutely. So if the concentration gradient switches, okay, many of these protein carriers will change the direction of movement. Okay, yeah, so some of them are like revolving doors that will move in either direction depending on the concentration gradient. Okay, so question to you. This slide, I've got this graph. I've got on the y-axis, I've got rate of diffusion, so going from 0 to increasing rates of diffusion. And on the x-axis, I've got concentration of substance that's being transported, okay? Going from 0 to increasing concentrations of substance being transported. So for something that is crossing the membrane, if it can cross the membrane, you would expect that as I increase the concentration on one side, that the rate of diffusion will increase, right? You agree? So as I increase the concentration, let's say on the outside of the cell, that the rate at which that substance moves in the cell is going to increase. Now, there are two transport mechanisms represented by two different lines. In the black line, that's the relationship that's being drawn, right? And it's just a straight line moving off into infinity. That as you increase the concentration, the rate of diffusion increases. But in the green line, starting at 0, as you increase the concentration, the rate increases until it hits some limiting factor, and the rate of diffusion is actually plateauing. So one of these represents simple diffusion, and one represents facilitated. Which one represents simple diffusion? Who thinks the black one? And who thinks the green? So the black line represents simple diffusion. Okay? That the more you increase the concentration, the more it crosses. Okay? The more transport. And the faster the rate of transport. On the other hand, facilitated diffusion is being represented by the green line, meaning that as the concentration of the substance increases, the rate of diffusion also increases, but then at some point you hit a limiting, you hit a really upper ceiling in terms of rate of diffusion. What's happening?
There's a limited number of protein channels.
There's a limited number of protein channels. that can carry this substance across. Okay? So this red line represents the maximal rate of diffusion that is going to be determined by the available transporters. At some point, you're going to saturate your transporters. All your transporters are running at maximum speed. They're all occupied. And you can't transport the substance any faster. So that is an important differentiating feature of facilitated diffusion versus simple diffusion. That you have a saturation point, okay? Because you have a limited number of transport proteins. Okay? The other feature, in addition to saturation, is specificity. Okay? That because the substance has to bind to a carrier protein, there is some specificity involved in that transport. Okay? You can't have any random substance binding to this protein. It's got to be that particular substance or something that's very similar to it. Okay? Does that make sense? Good. And when we talk about, this is actually something that becomes very clinically relevant when we get to the renal system and we talk about things like the reabsorption of glucose in the kidneys. So without getting too far into it, the way the kidneys work is we basically put everything that's in blood, anything that's dissolved in blood, minus the cells and the protein, we just dump that into the nephron, right? And then we start taking back everything we absolutely need, and everything else just gets eliminated in the urine, okay? So glucose, under normal circumstances, we take back all the glucose that was originally dumped into the nephrons. Okay? So that what's normal is that someone should not have glucose in their urine. But glucose is being transported by these transporters. So if someone has diabetes and their blood glucose quadruples, right? The concentration of glucose all of a sudden is too high. It maxes out the rate of transport, and you still have glucose trapped in the urine. Okay? So that becomes a real clinically relevant example to this difference in transport mechanisms. Yes, ma'am?
Do these carrier proteins carry across multiple kinds of substances, or in one substance?
They are usually fairly specific, but everything has sort of a preference for one thing and then like a relative affinity for other things. So it's possible for some things to sort of like masquerade as glucose and get across, but it's usually that transporters are very specific to their particular transport substance. Yes, ma'am?
What's the concentration needed so it's supposed to be the simple diffusion?
Say that again?
What's the concentration to get to where it's supposed to be the simple diffusion line?
So I see where you're trying to get, but what I'm doing is I'm constantly increasing the concentration. I'm maintaining that concentration gradient. That's what this is saying, right? That it's not like the previous slide where eventually it's going to equal out, because what it would be like is if I constantly added more here, right? Then we would have this continuous movement. Okay. Osmosis. Osmosis is, I'm gonna move on. I see your hands, so I'll solicit some questions in just a little bit. So osmosis is actually a form of simple diffusion. Okay? Osmosis is a form of simple diffusion, but it involves the movement of water across a semi-permeable membrane. Okay? And osmosis is one of these like deceptively simple concepts, right? That you think that you fully understand, and then like you get into it and it's like, oh, that's really interesting. It's more complicated than I thought. So the key features with osmosis is that you have two fluid compartments. You've got a semi-permeable membrane in between, okay? And the two fluid compartments are going to contain solutes. They're solutions. It's not pure water. They're solutions, and some of those solutes, this is critical, some of those solutes cannot cross the semi-permeable membrane. Okay? Osmotic pressure can only be exerted by a solute that cannot cross a membrane. Okay? Osmotic pressure can only be exerted by a solute that cannot cross the membrane. So if we're looking at the left side of the slide, so that we've got side 1 and side 2, if we look at these dark blue circles, we've got the light blue circles and the dark blue circles. The light blue circles can cross the membrane. Okay? Because they can cross the membrane, they're going to evenly distribute. They're going to diffuse, and then they're going to evenly distribute. Okay? No osmotic pressure. The dark blue circles are impermeable to the membrane, so they can't evenly distribute. Okay? So now you have two compartments with different concentrations. Okay? That's not gonna fly. All right? Because what water is going to do is, if the solutes can't move, then the water is going to move in order to equal the concentrations. But in order to equal the concentrations, it's got to get to different volumes. Does that make sense? In order for, so here on the side 1, we're starting off, we have 2 dark blue circles. On side 2, we've got 4 dark blue circles. The concentration of dark blue circles is higher in side 2. Okay? Another way of thinking about this is the water concentration is higher in side 1. Right? There is more water to solute in side 1. So in order to equal the concentrations, water has to move from side 1 to side 2 so that the solutes per volume, that's what concentration means, solutes per volume, unit of volume, is equal on both sides because the volumes are different. That make sense? So when we talk about osmotic pressure, these solutes on side 2, they exert an osmotic pressure. And osmotic pressures are pulling pressures. They pull water towards them. Okay? They pull water towards them. So these solutes that are impermeable to the membrane on side 2, because they are of higher number on side 2 per volume, they're going to pull water towards them until you have equal concentrations on both sides. All right? So where osmosis becomes very important, so there's a couple of terms. Osmosis is the phenomenon, right? And a way of thinking about osmosis, because it is analogous to simple diffusion. So simple diffusion, a substance was moving from high to low concentration, right? With osmosis, I know this isn't quite right, but you can think about it this way, is water is moving from an area of high water concentration to low water concentration, okay? That water is equaling out in concentration. Once it hits this level, there's no more pressure. Yeah, because now they are equal in concentration. Okay? So where this becomes very important is actually when we talk about tonicity, clinically, when we talk about tonicity. Because tonicity is the effect that a solution will have on cell volume. Okay? The term tonicity describes it's a property of a solution, and it describes the effect that that solution will have on cell volume. Okay? So, and things are described as being either isotonic. So if a solution is isotonic, iso means same. Okay? It has the same osmolarity, osmolality. Those terms are interchangeable to a great extent. It has the same osmolarity as the inside of a cell. An isotonic solution has the same osmolarity as the inside of the cell. Okay? As the same solute concentration. So what direction is water going to move? It's not, right? So quick drawings. The osmolarity of a cell is approximately 290, and the units on osmolarity are what's called milliosmoles. Okay? And osmolarity is actually a measure of the number of particles in a solution that can exert an osmotic pressure. Okay? The number of particles in a solution that can exert an osmotic pressure. An intracellular fluid has an osmolarity of about 290. So if this cell is bathed in a solution that is isotonic, what is the osmolarity of this solution? 290. Okay? No net movement of water. Okay? If the solution, on the other hand, is hypotonic. Hypo meaning less than, right? If it's hypotonic, it means that a hypotonic solution is a more dilute solution compared to intracellular fluid. It has fewer particles. So what's the number in relation to 290? Less than. Okay? So let's say it's 280. And this is something, when we get to the renal physiology, this becomes, again, a big deal, that as the numbers get smaller, the solution gets more dilute. Okay? More water, less particles. So this hypotonic solution is going to cause water to shift. In what direction? Out of the cell or into the cell? Into the cell. So hypotonic solutions cause cell swelling. Okay? Hypotonic solutions causes cell swelling. It causes this water to move into the cell. The cell gets big and bloated. Okay? Hypertonic solutions have an osmolarity greater than the cell. So let's say it's 295. The higher the number, the more concentrated the solution. Less water. What direction is water moving? Out of the cell. Hypertonic solutions causes cell shrinking. Okay? So where this becomes extremely important clinically? Where are my nurses? IV fluids, right? IV fluids, extremely important. And there's a wide variety of standard IV solutions. We've got the variety of isotonic solutions. What are some of the isotonic solutions, guys? Normal saline, lactate ringers, right? And even there's like the sort of isotonic, D5W, right? Dextrose water 5%. The reason why it's sort of isotonic, so dextrose water is basically sugar water. And it has the same osmolarity, but it's dextrose. It's a sugar. And the reason why it's sort of isotonic is because it goes in isotonic, but then what happens when you start absorbing that sugar and you start metabolizing that sugar? All of a sudden that solution becomes what? Hypotonic. Okay, because your particles are going away. Okay, so you've got normal saline, which is what, 0.9, I believe? Yeah. Normal saline, lactate ringers is a isotonic solution, and then D5W is an isotonic solution. So, and then you also have a variety of hypertonic solutions and hypotonic solutions that are appropriate depending on the clinical situation. But tonicity of solutions become extremely important when you're talking about IV fluids. Okay? Yes, ma'am.
When you were talking about the D5W, is that, and you said that you would absorb the sugar in it, does that happen every time you use it, or is it just the some?
No, you're going to absorb it no matter who you are, no matter what the situation, yeah.
It's only like isotonic.
Exactly, exactly. But there are situations where D5W is the appropriate solution to use. Yeah. Okay. Moving on. Okay, so active transport. Active transport, primary active transport, secondary active transport. Okay? Primary active transport, very straightforward. It uses the energy in ATP. Okay? And ATP is hydrolyzed, so it goes from adenosine triphosphate, so adenosine and 3 phosphates, to 1 phosphate comes off, and it's adenosine 2 phosphates plus 1. Okay? And that breaking of that bond releases energy, and you can do work with it. So probably the most important example of primary active transporter is the sodium-potassium pump, also known as sodium-potassium ATPase pump. And this ATPase refers to one of the subunits in the transporter. Sorry. That's gonna be great on the recording. One of the subunits on that protein is an enzyme, and it's an enzyme that's dedicated to hydrolyzing that ATP. Usually enzymes usually are written out as ending in ASE, right? ACE. So it refers to the enzymatic subunit in that protein. So the sodium-potassium ATPase pump is found in every cell in our body, and it's constantly working. It's constantly working. Something like over 50%, depending on the cell type, but over 50% of the energy of the cell is dedicated to running the sodium-potassium pumps. Okay? The sodium-potassium pumps, it pumps 3 potassiums out, 3 sodiums out, and then pulls 2 potassiums in. Okay? In one sort of go around, it kicks out 3 sodiums and pulls in 2 potassiums. And both of those ions are moving against the gradient. Okay? So this pump is absolutely critical for maintaining the ion concentration gradients between inside and out of sodium and potassium. Okay? So very important. Another form of active transport is vesicular transport, and I'm not going to talk too much about vesicular transport, but basically it is the movement of a substance into the cell or out of the cell through the formation or the incorporation of transport vesicles. So when a substance moves from inside the cell out of the cell, that process is called exocytosis. Okay? So let's say you are synthesizing a protein that, let's say the protein is a hormone, right? This is an endocrine organ. It's synthesizing its hormone, and it gets a signal from the outside to release that hormone. So that hormone is being stored in secretory vesicles, and they're just little bubbles of plasma membrane that contain something. Okay? That's what those vesicles are, little bubbles of plasma membrane. So this bubble of plasma membrane filled with hormone moves to the plasma membrane, and then the vesicle opens and it just incorporates into the plasma membrane and just dumps out the hormone. That's exocytosis. Okay? Something moves to the surface, the vesicle moves to the surface, incorporates into the membrane, and gets released. The opposite process is endocytosis, and that's when a substance that is in the outside, the extracellular space, gets internalized, right, and enters the cell by forming a vesicle and internalizing the vesicle. Okay? Now, endocytosis can happen in a variety of circumstances. It can involve a receptor, so the substance could bind to a receptor and then the entire thing gets internalized, or it can happen without the binding to a receptor. Okay, so it could be receptor mediated, receptor non-mediated. I'm not going to get into the details, but basically vesicular transport is another important example of primary active transport. Involves ATP. Secondary active transport is using energy, but it's not energy in the form of ATP. Okay? Secondary active transport is using the energy that is stored in the concentration gradients, predominantly the ion concentration gradients across the membrane. Okay? So what that means, let's take sodium. Sodium is an important one. Okay? Sodium has a higher concentration where? Outside the cell, right? So sodium has a higher concentration outside the cell, which means that it has a big motivation to get where? Inside the cell. Okay? So as long as sodium is sitting outside the cell and wants to get inside the cell, it's very similar to a boulder sitting at the top of a hill with a little wedge keeping it from coming down. Okay? So that's the little sodium boulder, and it really wants to go down. So the fact that it is in this position means that it has a lot of what we call potential energy. Okay? Now, let's say I take the wedge away. What happens to this? Sorry. Okay. Back to the boulder. I take the wedge away. What happens to that boulder? Starts screaming down that hill. All the potential energy gets transferred into what kind of energy? Kinetic. As all that energy is getting released into kinetic energy, if I was smart, and if I was smarter than I currently am, and could engineer something that could harness that kinetic energy, I could do work, right? And this is actually the basis for like hydroelectric power. You know, so you build like the Hoover Dam, and so all of this potential energy is jammed up against that little thumbnail of a dam, and then I just like open up a little spout and all this water comes rushing out. Okay? And I've engineered all of that I've engineered something that converts that movement of water out. It turns turbines and that creates electricity. Okay? So what secondary active transport is, you have these transporters that harness the kinetic energy released when sodium moves into the cell or some other ion moves down its concentration gradient, and it uses it to actively move a second substance against its transport, against its concentration gradient. So there's a lot of examples of these, and many of them use sodium because the sodium concentration gradient is significant and it's very well maintained. So sodium moves into the cell and we can actively pump then amino acids into the cell, right, against its concentration gradient. Or we can move calcium out of the cell. Okay? But the nature of the structure of this protein is what's actually engineering the harnessing of that energy and the conversion of it into something that does work. Okay? So in secondary active transport, there's always two substances moving. There's always at least two substances moving. One of them is moving downhill in terms of its concentration gradient. And the other substance is moving uphill. Okay? So secondary active transport, there's always at least two substances moving. One is like the helper substance, like sodium, and the other is the actual substance of transport. All right? So examples, so no matter what's happening, these two substances are always moving in opposite directions with respect to their concentration gradient. Okay? So you might want to write that down. That's important. The two substances with secondary active transport, they are always moving in opposite directions with respect to their concentration gradients. Okay? With that said, if you're looking at whether they're moving into the cell or out of the cell, sometimes they're both moving in, sometimes they're both moving out, and sometimes they're moving in different directions. Okay? So when two substances are moving in the same direction with respect to extracellular and intracellular movement, we call those co-transporters. Okay? So these are examples of co-transporters. And if you notice, there's a lot of them that use sodium. Bless you. That's not actually what you say when somebody hiccups, but so a lot of them involve sodium. So in every single example of these co-transporters, what direction is sodium moving in? Into the cell, right? Because that's downhill for the cell, for sodium. That's the direction of movement. And sodium is a great workhorse. That's what it does. It moves into the cell, it frees up energy. But for the rest of these that are also moving into the cell, that just happens to be the... Is that a baseball game? All right. I can't compete with a baseball game. So for these co-transporters that are using sodium, we know sodium is the workhorse, and we know sodium is moving into the cell. The other substance that's moving into the cell is moving against its concentration gradient. But its concentration gradient happens to just, against the concentration gradient, happens to also be pointing inside the cell. Yeah. To their concentration gradient, that's absolutely right. So, two substances, they're always moving in opposite directions with respect to their concentration gradient. One's moving downhill, the other one's moving uphill. Yes, 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 that's what I meant. Is that what yeah. That they're moving in opposite directions with respect to each other. So one is moving uphill, the other one's moving downhill. Yeah. Okay. Co-transporters, they're both moving into the cell or they're both moving out of the cell. With exchangers, I know it makes no sense right now. Believe me, when you're not at, I promise you when you wake up in the morning it will make more sense. You've been in class for like 13 hours or something crazy, all right? Exchangers, they are moving in opposite directions with respect to into the cell or out of the cell, all right? This is always a topic that like is just a little bit, takes up just a little bit of time to kind of sort through in your brain. All right, but I promise you it will make more sense. Okay. All right, so I have kind of one last major idea I wanted to get through before we finished today. And this is, so every class that teaches physiology, every physiology textbook or class always talks about homeostasis, right? And homeostasis is just one of those topics that, like, is so much better a concept than it is ever explained in any book, right? It just makes you want to shoot yourself sometimes how poorly it is talked about. So, so this idea of homeostasis, it's really an idea that was groundbreaking when it came out. Okay, it was first, I want to say Walter Cannon. He wrote a book. He was one of the first physiologists. So he wrote a book about homeostasis. And it was groundbreaking because up until that point, what we really talked about in science was based on the physical sciences, and it was based on equilibrium, right? Getting to some chemical equilibrium. And the reality is that the body is not in equilibrium. In fact, at a cellular level, equilibrium is death for cells, okay? Cells die, and then they get into equilibrium. So being alive to a certain extent means that there's this tension. You're actually not in equilibrium. Because that tension, there's energy stored in it, right? The fact that sodium is constantly trying to get in the cell it's the Hoover Dam. It creates all of this energy, this tension that keeps your cells alive. It allows it to do work and keeps it alive. So homeostasis is this idea that replaced the thought of equilibrium in terms of biological systems and really refers to stability. Okay? Homeostasis really refers to stability in the system, okay? Which does not always include, often doesn't include equilibrium. Okay? So we have our basic generic cell with all its basic generic components. And then we have developed these specialized cells that have beefed up some of these basic components with these basic functions. So we've got muscle cells, right? Myocytes. And they include skeletal muscle and cardiac muscle and smooth muscle. And these cells have become specialized for intracellular movement. So all of these proteins that make up the cytoskeleton in a generic cell have been elaborated, right? These actin and myosin fibers have become greatly elaborated so that you can generate significant intracellular movement and movement of limbs and generation of force, right? So we have this specialization of muscle tissue. We've got nervous tissue. Nervous tissue, the specialization with nervous tissue is in its ability to fire what we call action potentials, which are electrical impulses that move from one end of the cell to the next. And that's all about the plasma membrane and the distribution and the density of ion channels that we see at the plasma membrane. Okay? Epithelial tissue. Epithelial tissue, what we're specialized for with epithelial tissue is selective movement across the plasma membrane. Right? It's another cell where the plasma membrane is what's the key difference. Okay? Epithelial tissue forms the barriers in our body. Anywhere you have a barrier between the inside of you and the outside of you, you have epithelial tissue forming that barrier. Because epithelial cells are tightly connected to one another, and anything that wants to cross from outside to inside has to cross 2 layers of plasma membrane. Okay? And only certain things are allowed to get through. So we have epithelial tissue. And then super specialized epithelial tissue in the form of glandular tissue. Okay? Glandular tissue is epithelial cells that have evolved to produce either a hormone, in the case of endocrine glands, where the substance is directly released into the bloodstream, and we have exocrine glands where the substance that's being produced, we don't call it a hormone when it's an exocrine gland. It's being released onto the surface of an organ. Okay? So many times what's super specialized in this, the function that's being beefed up in this tissue type, is actually protein synthesis. So lots of ribosomes, lots of endoplasmic reticuli, lots of Golgi apparati, right? Complexes. Okay? Lots of protein synthesis machinery, because that's the function of this, is to synthesize protein. So we have this interdependence between the cells that really are the basic units, the tissue types that they form, the organs and the organ systems, okay, that maintain the health of the organism as a whole, but then that organization in turn maintains the health of the cell. Okay? So homeostasis is stability. And one of the most important things that we need to remain stable is extracellular fluid. Okay? Because extracellular fluid is the environment, the immediate environment of all of our cells. We have to keep a stable, healthy composition of extracellular fluid in order to keep healthy, stable cells, functioning cells. Okay? So there are a wide variety of variables or parameters of extracellular fluid that is very tightly regulated. And we talked about 2 of them, right? Temperature and pH, extremely important. And when we talk about extracellular fluid, clinically, we're talking about plasma. Okay? Because that's the extracellular fluid that we generally have available to us to evaluate, is plasma. So we can say plasma for all of these things. What's the plasma concentration of glucose? Are you hypoglycemic? Are you hyperglycemic? You have normal glucose levels? Well, we have to make sure that they're normal. Okay? That's tightly regulated. Oxygen, carbon dioxide levels. Are you hypoxic? Is your oxygen concentration low? Are you hypercapnic? Is your carbon dioxide too high? Okay? That's got to be controlled. your waste concentration. Is your uric acid level too high because you have gout? Okay, because you can't break down uric acid effectively? Okay, that's being controlled. Water concentration in the form of osmolarity is controlled. Our electrolyte concentration. Okay? So for all of these variables, we have a set point or a range that is optimal, that represents stability. Okay? Some sort of set point or range that represents stability or homeostasis. Okay? And the way that we maintain stability is through what we call homeostatic control systems. Okay? And the very next unit for us is control systems. Okay? So homeostatic control systems utilize either the nervous system, the endocrine system, or both. Okay? And these homeostatic control systems are constantly monitoring all of these parameters. And if something gets too high, something kicks in order to bring it back down. And if something gets too low, something kicks in to bring it back up. Okay? That's the homeostatic control system. So nearly every textbook that you will pick up is going to show you the same basic structure for a homeostatic control system. Okay? It's going to show you something that looks like this. You have some controlled variable, let's say blood glucose. Okay? And the controlled variable, there's sensors that are sensing glucose concentration. Okay? And these sensors are informing some sort of control center, oftentimes the brain, about the status of glucose concentration. Let's switch it. Let's say body temperature. Okay, let's say that the controlled variable is body temperature. And the control center is the brain. Your thermostat is in your brain. And your brain is constantly reading body temperature and comparing it to the set point. And if it deviates from that, it's going to stimulate some sort of effector, like skeletal muscle, and make you shiver if you're too cold, in order to generate the heat that brings about the return of the control system to the set point. So every homeostatic system is, the vast majority of homeostatic systems are described as being very simple negative feedback loops. Okay? And they even use the thermostat example oftentimes to illustrate simple negative feedback loop. Okay? So negative feedback loop as applied to the room temperature example. So if this was in the dead of winter, this would be a little bit more relevant. But you set your thermostat to a nice 72. You like it warm. You like to sit around in shorts and be comfortable. So you set the room to 72, and it's really cold outside, so the room falls to 60. So what happens is that the thermostat in your system is going to read that temperature change through the thermometer, and it's going to turn on the furnace to increase heat output to bring the room back to 72. And then as soon as that thermostat reads that the room is back to 72, it's going to shut off the furnace. Right? Simple. Negative feedback loop. Right? But if your heating system, and if you live in New York City, this is probably true, if your heating system worked like this, your house would either always be incredibly hot or incredibly cold. Okay? Because this system doesn't work, and it's not how your body works. Okay? Because how much time do you think it takes for a thermometer to get an accurate reading of room temperature? A minute? 2 minutes? An hour? That might be a little too much, but let's be real. Let's say it's an awesome thermometer. Let's say it takes 3 minutes. okay? 3 minutes. How long do you think it takes the thermostat, the mechanism of the thermostat, to read that change in room temperature and turn on the furnace? Let's say another 3 minutes, okay? We're at 6 minutes now. Now this is the big thing. How long do you think it takes the furnace to go from cold to turned on and producing enough heat to change room temperature? That might be like 20, 30 minutes, right? So now we're at, let's say, 30 minutes in total. 6 minutes from before, 24 minutes for the furnace. So let's say we're taking 30 minutes to get from here to here. That means that the thermostat is always chasing its tail, right? By the time it registers that the room temperature has gotten back to 72, it's going to take a half hour for things to come round again and for it to shut off. So a system like this doesn't create constancy, right? It doesn't create stability. It creates oscillations, overshoots and undershoots around the set point because there's a time lag. All right? So the missing part of all these systems that actually exists in your body is some degree of prediction. And that's what's true about your more sophisticated if you're comfortable when you get home, then you have a thermostat that knows, okay, 60 degrees, we want 72, I have to turn on the furnace for 10 minutes and then shut it off to bring it up to that number. It has to be smarter. It has to be able to predict how long to turn on the furnace or how much stimulation to the system. And then it can use the feedback that it gets to validate its prediction or validate its assumption. Okay? And so if we actually look just for on a very superficial level of how the body actually controls body temperature, it's much smarter than we've ever been led to believe, right? So your thermostat is in your brain. It sits in your hypothalamus. And it actually is reading 2 temperatures all the time, 2 very different temperatures. It's reading core temperature, and it's reading skin temperature. Okay? Now, when we talk about maintaining body temperature, we're talking about the core. Your skin temperature is always going up and down, right? It all depends on what's going on in the environment. But what the brain is trying to do is maintain core temperature. And so in a very basic way, the brain is using the information from skin temperature to predict whether it needs to turn on the furnace or shut off the furnace, so to speak, in order to protect core temperature. So the information it gets from skin temperature is its prediction, okay, is at least one level of its prediction in order to maintain core temperature. Does that make sense? So one thing about these homeostatic control systems is that they all include some degree of prediction, and they use different mechanisms to get that prediction so that they are smarter systems, so that you're not just oscillating around the set point, that you're actually achieving it. Okay? So I know you're exhausted.